Okay, and I will share the screen. Here we go. Okay, let's get rid of this and that, and go to, this thing's a pain. All right, so, and I'll make that go away for right now. Okay, so welcome to the first lab. It's not going to be a long lab, especially once you get going. You learn how to use the blood glucose meters. But before I go over that, I want to go over lab safety. There's no open flames. We don't have dissection scalpels. We don't have, well, we do sometimes have glassware, but we don't use any obnoxious chemicals like have animals preserved and fixative and stuff like that. So the only thing that could really happen is just make sure you put your backpacks, whatever, under the table or someplace out of the aisles, because I and you guys are going to be walking between them. And if you trip over a backpack, this concrete floor is pretty unforgiving. So that's one thing that can happen. The other thing, and I have to go through it, and that's what the chemical hygiene is about. I know you signed them before from other labs. It's just, I'm going to turn it into human resources, and then I'll run over safety procedures in the lab. If you should find yourself on fire, again, no smoking in the lab. Here's the fire extinguisher, all right. Should you find you get some chemicals in your eye now to lab? One time I was using, because when COVID was really bad, I was using Lysol on the discs. And I washed my hand, but I barely didn't wash enough, and then I rubbed my eye, and I got Lysol residue in my cut. I just went to a doctor. But should you get something that's burning in your eye, let me know for one thing. And here's the eye wash station. You just face your eyes open and press the lever. All right. Should you take a bunch of Lysol or bleach and it didn't spill all over your body, here's a full body wash station. Just make sure it's an emergency, because once you pull the lever, it's an all or none response and the water's going to come down. Other than that, each lab that we have, I think there's some sort of health or safety concern, I'll bring it up that day. Like I have some really good ones here with this blood glucose lab that I'm going to go over with you, all right? Should you ever have a lab come up that you have a health concern about, please let me know, okay? And you can be excused from doing that lab, because like we do blood pressure, measuring blood pressure. I've had students that are on high blood pressure medication, or they have what's called orthostatic intolerance, which means they don't tolerate changing body postures very quickly without passing out. So if anything ever comes up that you're concerned about, let me know, all right? It's just a lie. It's not meant to cause any harm. Okay, with that being said, I'm always going to have fun. I'm just getting going. You just want to safety stuff.
Yeah, I'll say that.
Oh, you want I talked a lot of so I always put on the left-hand side of the board what's going on this week. So lab safety, check. Then we'll start the blood glucose lab. And then this will be the back of the lab report based on. Next week is one. Come to class on time. Okay? Because I hand out the quizzes that go through. If you come in late, and other students are taking the quiz, it's up to me to say, okay, we'll reschedule this, and you must take the quiz within 1 week or you get it through. And you have to let me know what's going on. Okay, so now that you know where the lab is, try to come on time when you have a quiz. In general, come on time. Here's how the quiz is going to be. They're going to ask what you're going to cover in the quiz. Well, here is our handout for today. All right. Look at all those nice bold-faced vocabulary terms. Fortunately, a lot of it we've already covered in a lecture on metabolism, including diabetes. So there's a lot there. What we're doing today is called an oral glucose tolerance test. I could ask you on the quiz, what does the OGTT stand for in this lab? And it's right there. So know your bold-faced vocab words. And you're also going to want to know the procedure, okay? It's very simple. I'm not going to go into detail. So there's going to be 10 questions next week on this lab. Bold-faced vocab terms, maybe what we did, and also I could ask you what the data told us. Not your individual data, but it's going to be class data. So I could ask for the people who drank the sugar solution, what happened to their blood glucose, average blood glucose at time 30? You don't have to know the number, but you have to know if it went up, down, or stayed the same. That's what you're going to expect. Now, if I go back, so that's 10 questions. There's going to be 5 questions on the lab that you're coming next week to do. And there will be 5 questions on bold-faced vocabulary. So if I pull up that lab handout, make it a little bigger for you, all those boldface vocabulary words. I can ask 5 questions on that, okay? Every quiz will be like that in lab. 10 questions on the previous week's lab and 5 questions on the boldface vocabulary of the lab you're coming in to do. So do try and look at the handout next week with time enough to study those terms. Good question. Any kind of question. Multiple choice, matching, fill in the blank, short answer, okay? Since we're going to be covering how the body regulates blood glucose through negative feedback, I certainly could ask you, I know that insulin lowers blood glucose, but how does it do it? And that would be a short answer. And then, because you've been studying for the first quiz, you'd say, Dr. Who? Because insulin tells the cells to take in glucose from the bloodstream. That's how it works. All right, that would be a short answer. And that would be a very good question to ask. Any questions on the lab quiz? Okay, so I think I'll leave that up. Okay, so here is the procedure. Well, I'm going to go over how you use the blood glucose meter. How many people know how to use a blood glucose meter, the lancet and the lancet holder, to get a blood glucose reading? No one. Don't worry, it's super easy. But let me say this. These are not good / expensive glucose meters. Neither are glucose test strips. If I were a diabetic, I'd buy higher quality equipment to work with. These are bargain basement, okay? So if you see blood glucose readings of 60, say, oh my God, Dr. Barbeau, you know, am I going to pass out? No, if I took 2 readings back to back, I'd get slightly different readings from this meter. But it'll work well enough for the experiment. Okay, so I'm going to ask, and you're going to fill in because you've been studying for this first quiz, right? What blood glucose is regulated in negative feedback? This is what you're doing. So if blood glucose rises too high, that's a stimulus. It's outside of the normal range. What is the sensor, integrated sensor, and effector that senses it, understands it's outside of the normal range, and then will secrete a hormone to fix it? And I couldn't help blood glucose. That would be for temperature or oxytocin release. So now, nice and loud because it came through the fan. Pancreas, yes. Pancreas. And to bring blood glucose back down, it secretes insulin. And of course, you know from what we discussed in lecture, insulin tells your cells to take up that blood glucose and that thing gets lower. The opposite, so this is what's going to happen to people who drink a full sugar drink during class today. If blood glucose drops too low, the same sensor, integrating sensor, and effector responds, but now with this, it secretes. Moving on. And how does glucagon make your blood glucose go up? Nice and loud and confident. First of all, what organ is going to be responding to this glucagon? The liver. And the liver is going to break down what? Glycogen. And then release free glucose into the bloodstream, and that makes the blood glucose go up. I could ask that question next week, lab quiz, or the lecture quiz. How does glucagon make blood glucose go up? Then you have to put it in the parts. Okay, now that you've got this down, in the lab report, when I asked you to discuss how blood glucose is regulated through negative feedback, right there, okay? All right, so here's what you're going to do. You're going to break up evenly. I hope I'm not. I've got more cups than I need, but I got a pack of you, okay? I said 14. 7 people should drink the full sugar drink. Here's what it is. I'm going to have one that's going to be one over on Thursday. And it's not good sweet tea. It's not your mama's sweet tea, okay? It is food line iced tea mix. Okay, and then here is just plain water. It's room temperature because you don't want to drink it fast. You don't put this cold and up. Okay, so you're going to take your beverage. You're going to drink it. Roll the cup out. Set your timer. Calm for 30 minutes. Then we're going to sit down and give a time 0 blood glucose reading. The reason you drink it and time it first is going to take 30 minutes from when you drink that solution to go through your stomach and to get into your intestines and get into your bloodstream. Okay? So during that 30-minute time, it's not going to take 30 minutes. You're going to measure your blood glucose and rate it into an Excel spreadsheet with I'll show you. After the timer goes off, you take your time 30 blood glucose reading, set your timer for the last 30 minutes, and then when that comes up, record your time 60 blood glucose reading. Okay? We're not going to go to 90 minutes. We should see the effect of insulin by time 60. So here's what we expect. The people who drink a sugar beverage, at time 30, your blood glucose will go off, right? Just like it would if you ate. Then, when your pancreas senses the blood glucose went up, it secretes insulin at time 30, and 30 minutes later, insulin has its effects and your blood glucose should come back down. That's how fast it is. People who drink water, your blood glucose shouldn't change over time because you didn't consume any sugar. And that's what we expect, right? So, here's what the spreadsheet looks like, and then I'll show you how to use the lancet and blood glucose meter. So, trying to get the point. The top table is blood glucose after the sugar drink. So if you drank the tea, that's your table. And simply pick a line like line 1 and just keep putting your data through the whole experiment. If you drank water, you're going to be in the gray table. And so put your data all in one row there. Excel will automatically, because I put a formula in, calculate the average blood glucose at each time interval right here. And then I'll have a t-test comparing time 0 and time 30, and then time 30 and time 60. The intervals I'm really caring about are time 30 and time 60. At time 0, I don't know what you ate or drank before coming into lab. That data is kind of going to be sloppy, all right? But between 30 and 60, if we see a difference, that's going to tell us something. Okay, so as soon as you get your data, put it into Excel so at the end of lab, there's not a long line of people trying to get on the computer. Okay, I have at your table lancet holders that are in the open position. You might have these blue ones. You might have this light blue and white one, shorter. And then some of you, I think the back table might have this black one with a clear top. Okay? But notice the cap is off of all of them. This is serious safety stuff right here for lab and in your future career as nurses. If you see a lancet holder in the closed position, it's like picking up a gun. You don't know if it's loaded or not. You don't know if there's a dirty needle in here, okay? And if it's cocked and ready to go and you grab it haphazardly and it goes off and sticks you, you're gonna have to go to human resources and get treated for viral problems and things like that, okay? So it should always be in the open position. When you leave lab today, they better be in an open position on your desk because that's a big no-no. I need to know, and tomorrow's class needs to know that the chamber is open. Okay, so, could you hand me that purple box of lancets? Okay, so let me get back to this. Share. Get rid of that. Get rid of that. Okay, where's my screen? It's not showing. Okay, how about I stop the share and then I go back to share. No, actually, I'll do this right here. This is just for the people who aren't here today to see what it is that we're doing. So it's got a little sterile cap on it that you must twist off, okay? So it can expose the needle. I'm gonna use this one right here. You put the needle into the holder. You don't have to jam it in there too hard. You're gonna put the cap back on, screw it on, and then it's at the top, you kind of want to turn it clockwise to the full position because you want the needle to come out far enough to make contact with the skin. Then I'm just grabbing one of these alcohol swabs from a box over there, but I've got hydrogen peroxide and cotton balls for you that you can use to clean your finger. Okay, so I've got that out. Could you pass me the glucose meter in that that little black pack there? Yeah. Okay. So the glucose meters work. Oh, and thank you. And the glucose test strip. Thank you very much. Okay. Don't put the glucose test strip until you've got blood ready to go in your finger, because the glucose meter, okay, will turn off after about a minute. And if you're struggling to get the blood drop on and then you turn the blood glucose, it's too late. It's timed out. All right. So I'm just going to keep it here. The glucose test strip, the gray tab that has the little white lines, go into the glucose meter and it will automatically turn on. The other side of the glucose strip is white with a yellow chamber. That is going to receive the blood. And you don't put the blood on top of the strip because it's got plastic coating. You put the strip, the yellow chamber, up to the blood drop and it'll suck it up by capillary action. That's how you charge the strip. Okay, so I'll clean my finger. And I'm telling you, I can't tell you how many times I've done this to demonstrate it to students. It does not hurt. It's 28 gauge. It's small. Okay? So you cock it like a gun. I'm going to make sure it goes. That one didn't, I didn't cock it enough. Then when you hit the button, the needle goes faster than your eye can perceive, like that, and pokes you. So you got to cock it, and then it's charged and ready to go. Okay, so I've got my glucose test strip. I got everything ready to go. I'm going to go ahead. I didn't cock it loud enough, okay? Oh, that hurts so much. It really doesn't. I'm going to squeeze a good drop of blood. Okay, now I'm going to put the test strip in the meter. It'll turn on automatically. And when you see little blood drops going across the screen, it is ready to go. It's ready to go. Put it up to the yellow channel. It sucked it up. Let me get my little swab here. 5, 4, 3, 2, 1. 1:55. I just ate lunch. Okay, so I expected it, well, 1:22 backwards when I show it on that. Okay, so you just put the strip up to the blood drop. It times off. You get your reading. Write it down before you forget it. Now, before you do anything, I now have a plastic glucose test strip and a little bit of blood on my swab, okay? That can go in the hazard trash down here. But it's not gonna poke a hole in a plastic bag and stick somewhere. However, the needle that I just used has been used. That goes into the hard plastic waste receptacle because they won't be able to poke through that plastic. So you do that before anything, and then you put your lancet down in the open position, you know you're good, okay? So, I don't think if there's anything else I've got to do. I think that's about it. Everyone understand your assignment. Half people choose the sweet drink, half choose the water, set the timer for 30 minutes, and sit down with your glucose meter. And I now have some extra lancets if you need them, because there's only 2 per table. All right? Go for it. Recording. This is for the benefit of our quarantine students. Okay, so on the syllabus, the online syllabus, it is click here for grading guidelines. I showed you in class today. So when you pull that up, you get this. It's thinking. There we go. Make it bigger. Can you see this with the lights on enough? Okay, so it's worth 35 points, which is not a hell of a lot, okay? And you can't go wrong if you follow the guidelines. It's like, this is why I made it. So you want to, and spelling and grammar count. So don't write like you text. You want to give it a title. So you don't just want to put blood glucose lab report. Put a little more to it, okay? Changes in blood glucose over time after consuming different beverages, something like that, okay? And then after that, you're going to put your name, Department of Biology, Francis Marion University, Florence, South Carolina, all right? And I'll show you where I have an example lab report for you to look at. Abstract, it comes at, and you don't have to have separate pages, like title pages, keep it all together. Then you're gonna have an abstract. It's best to have completed all the other sections of the lab report, then write the abstract, because an abstract is a summary sentence or 2 of all the sections that come after, all right? But basically, you're going to get 1 point for summarizing what was done. Examine changes in blood glucose at times 0, 30, and 60 after drinking either sugar-free beverage or full sugar beverage. I can't make it any more simple for you. Do try and put it in your own words, all right? I'll recognize if you just copied and pasted from my guidelines, okay? Don't do that. 1 point for summarizing your findings. We found, and this is what we expect, that blood glucose increased at time 30, but decreased at time 60 for the sugar beverage drinkers. And blood glucose did not change over time for the water drinkers. So that's it, real straightforward. The intro, this is where you want to have that negative feedback regulation of blood glucose, the full thing, all right? So 2.5 points, I'm trying to get my cursor. For what happens when blood glucose is too low, I did the opposite. Sensor, integrating center, effector, what is secreted, what it does, and then 2.5 points for when blood glucose is too high, and the full thing. And then after that paraphrasing of the negative feedback regulation of blood glucose, you want to cite your reference, okay, where you got that from. Where did you get that from? Let's go back to the syllabus, and let's go to our textbook, because you want to cite a source, a textbook or a publication. So here is the Human Physiology Wikibooks, and thinking. But the wireless connection in this room is not the best. Okay, so it was chapter 1, and it was wherever I talked about negative feedback, okay? So that's going to be page 5, okay? That's where I give, in the comment sections on the right, if you had this on your desktop or whatever, that's where I talk about, and they talk about negative feedback, page 5. Now, you don't put page 5 down. What you do is, let me go back to this. I'm sorry, I'm going so many places. I'm going back to general guidelines on grading a lab report. I have a practice or example lab report for you prepared. So I'm looking for, okay, click here for guidelines, click here to see an example. Yeah, so we want this one. Nope, not that one. I'm sorry. I thought I had another one. Let me just get rid of some of this stuff. Okay. Okay, go back to the syllabus. I must have closed it. Just want to show you where it is. If I can't find it and you can't find it, right? Click here. Okay, here it is. Click here for, I can't see my cursor. There it is. You don't really need to pay attention to all the gobbledygook up there unless you want to. But if you scroll down to this where it says example lab report, here. So this is an example. I did it on osmosis across an egg cell membrane. Title, name, abstract, introduction, osmosis, blah, 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 blah. And then over here, I have a superscript number 1. That's saying to the reader, go to reference number 1, which ends up being listed in the lit cited section. And that means that's this source right here, okay? You must have at least the wiki textbook as a source. You can use any other sources you want, but that's a requirement, okay? So after your negative feedback regulation of blood glucose, you put a superscript 1, and when I cruise down to your literature cited, number 1 is your reference. Okay, then you want to give a purpose of the experiment. Real easy. In this experiment, we wanted to determine if blood glucose changes over time in subjects who drank either a full sugar beverage or a sugar-free beverage. That's a purpose. Hypothesis. I already told you what we expect to happen. If you consume sugar, it should change your blood glucose over time. If you don't consume sugar, your blood glucose shouldn't change, certainly not in an hour. 1 point for including a properly cited numerical reference, which I just showed you how to do. Okay, methods. 1 point for including total number of people in each group. So when I look at all the data, when it's all entered, I'll look at how many were in the sugar group and how many were in the water group. 5 points for including the design. So simple. Measured blood glucose, milligrams per deciliter, using a 28 gauge lancet and lancet holder with glucose test strip and a glucose blood meter. And the glucose was measured at 0, 30, and 60 minutes. All right, that's it. 1 point for including the beverages and volume consumed. That's on the board. 350 ml of the sweet tea or 350 ml of water. And then 1 point for stating that the data was entered into Excel and summarized, and data was analyzed using t-tests. And I put that on the board as well. Results, you're simply going to report all your numbers, okay? So if you look at our data sheet, you're going to report average blood glucose at time 0, 30, and 60 for the sugar drinkers, and the same 3 average numbers for the water drinkers. You're also going to state for the t-test p-values, okay? So you're going to have basically 2 p-values. You're going to have right here 0 versus 30. That's comparing this column with that column. And then the big one is 30 versus 60. That p-value is going to be there. Now, I don't want to put it on the board, I don't need to. In general, if your p-value is something less than 0.05, like it's 0.01 or 0.025, something like that, okay? If it's very small, less than 0.0, those two columns of data are significantly different, and you can say one is larger or smaller than the other. If the p-value is larger than 0.05, like 0.06 or 0.09, or that's saying the two columns of data are not statistically different, so there's no difference between them. Okay, so back to, so you got it, it's numbers only. Don't explain the data, just averages, p-values, then state this went up at time this, and this went down at time that, and this did not change over time for this group. Now, for the reason, and it's all here. Everything you got to put down is explained all there. Okay. Discussion. Here's where you explain what the data means. If blood glucose went up at time 30 for the sugar drinkers, it's because they just consumed sugar. And 30 minutes later, wow, it's in their bloodstream, okay? But you need to explain why it went down at time 60. And it went down at time 60 because what happened at time 30, the pancreas secreted insulin because it sensed your blood glucose was high, and 30 minutes later, insulin had its effects and lowered the blood glucose. Then you also must explain why would blood glucose not change over time for the water drinkers? They didn't consume any sugar, so it shouldn't change. Then you must also end 2 points here for a possible source of experimental error. Can you think of one possible source of experimental error? Huh? Having eaten or drank something before this experiment, okay? Another good one is maybe the blood glucose meters and test strips weren't the most accurate equipment to use, all right? And that is a true thing. I do know that from these, okay? And then you have your full literature cited section, which I even show you how to cite the wiki textbook right here. You can copy and paste that and then put in page 5. That's it. That's the lab report. All right. It won't be due until, I think for you guys, it's February 14th. Let's look. I don't need that. It'll stay right here online. Oh, I think I made it due Friday, February the 14th. So that'll be in lecture. All right. Now notice that gives you plenty of time, okay? Because this is this week, then this is next week, and this is the following week, and this is the week of February 14th. So this week, Friday, is when that lab report is due. We also have a lecture exam that day, so make sure and have that lab report done early. Because if you get a rough draft together, even as soon as next week or the week after, I'll look at it in lab. I'll look at your rough draft. I'm not going to check spelling and error, but I'll see if something's missing from it. So if you get a draft done, get it to me early, then you can get your lab report done early, and you won't be trying to do a lab report and study for an exam at the same time, which is suicide. Okay, try not to do that. That's it. Yeah. Yes, if you want to do it on paper. I prefer paper submissions because honestly, it's easier on my eyes to grade paper copies. Yeah, much easier. It's so much more difficult to edit a PDF, and you know, that's, plus my eyes get all googly after looking at 30 or more lab reports on a computer screen. That doesn't work well for my eyes, okay? Any other questions about that or where everything is that you need? Awesome, okay. All right, so I'm just going to continue. Pause. Okay, so we're going to continue. The students have actually left the lab, but I do want to go over the data for my students who are still quarantining at home. So what we've got for our data looks like this. Okay, so the table up top with the black bar is the blood glucose levels over time after having drank a sugary beverage. And so we have a sample size of 5 students. The average at time 30 was 85.2, at 30, 137, and at 60, 107.8. Now the t-test compares, as you can see, the blue bar and the pink bar, red bar. I'm comparing those two columns, and that p-value is a very small value, 0.004. As long as the value is less than 0.05, you can say with confidence that those two data sets are different and that time 30's average was much higher than time 0. When I compared time intervals 30 and 60, my p-value there is 0.019. That is also very small. And so you can say with confidence that the average at time 60 was lower than the average at time 30. And this is what we expected to see. What I've done in the chart is I've put letters to indicate significant differences. So here for time 0, it's a letter A over the bar, but at time 30, it's a letter B because those two data sets are different from each other. Similar at time 60, I gave it a letter A because that makes it different from time 30. Okay, now for the group that drank the sugar-free beverage, the water, that's the table down here in gray, we had 7 people in that group, and the t-test p-value comparing time 0 and 30 was 0.083. That's above 0.05. That means I cannot say that the time 0 and time 30 groups are different from each other. They're the same. Likewise, comparing time 30 and 60, I have a p-value of 0.055. That would round up to 0.06. That's not significant either. So it did not change blood glucose over time for the water drinkers. And that is why in the chart, they all have the same letter, indicating they're all statistically similar to each other. Okay. So no one's here. I don't have to wear my mask. So I will make a short YouTube video for my students who are quarantining about how to do the lab report based on this particular lab. But this video is just going to be for what we did during lab. So I'm going to go ahead and stop recording and post this.